scar tissue suppresses the formation of the healthy tissue. So in regenerative medicine, the big goal right now is to try to figure out how to suppress the formation of scar tissue. It's true in muscle regeneration, it's true in nervous regeneration especially. Once you form scar tissue, you won't regenerate the nervous tissue. But if you can suppress that, turns out nerves and muscles know how to regrow pretty well. So we're seeing some advances in that. Like today, you don't have to look in the news too hard to be able to find examples of people that have had severe spinal cord injuries, and if they received immediate cryotherapy, where they cool the injury down and they inject some chemicals to try to prevent the release of pro-inflammatory compounds, it'll actually re-heal. It'll heal. There are some football players that had damaged spinal cords that received immediate treatment on the field, and they're walking now, whereas 10 years before they'd be paralyzed. So that's good. You know. Unfortunately, the emergency room is usually 15, 20, 30 minutes removed from when the injury actually takes place. So by the time you get there, it doesn't have as much of an impact. It has to kind of be at the point of injury. So we'll see. We'll see how that advances. But I worked with a guy at Ohio State who spent his whole life working on regenerative medicine. And, that's probably the most advanced we've seen in 50 years working on regenerative medicine. So who knows? 10 years from now, what they'll be. Angina pectoris is just chest pain associated with mild ischemia of the heart. It means that the heart muscle is not getting oxygenated properly, and as a result, it has spasmic contractions, and that winds up being felt as chest pain. Angina pectoris is called. Can be an indicator of an impending heart attack. So here's the part we didn't quite get to in lab. That's a histology section of cardiac muscle cells. So we'll just revisit a few general properties of muscle cells, and I think you guys seem to have kept a pretty good handle on it. Muscle cells are contractile. They're the only contractile tissue in the body. They're excitable, which means they can propagate action potentials. Okay. Then we get to the different types of muscle tissue, which vary a little bit. Striated muscle tissue, which includes skeletal and cardiac, and then smooth muscle tissue. Skeletal muscle is the one we spend the most time talking about in the EMT1. Right? We talk about the sarcoplasmic reticulum and calcium release and all that fun stuff. And then we don't spend a whole lot of time talking about smooth muscle and cardiac muscle. Here we'll talk about cardiac muscle. So some things that set cardiac muscle apart from skeletal muscle, and some things that are the same. Okay? Cardiac muscle and skeletal muscle are both striated tissues. So these striations, I don't know how well it shows up here, but you can see those little marks going across there. Okay? Those are striations, which maybe you might remember are the A and I bands, where the actin and myosin are overlapping. Okay? And that's what makes those striations. The sarcomeres have to be nice and regular in order to do that, and so in skeletal muscle and cardiac muscle they are, but in smooth muscle they're irregular, so you don't see striations. Cardiac muscle cells are much shorter than skeletal muscle cells. Skeletal muscle cells are hundreds of micrometers, maybe even centimeters long. Okay, they can be very long. Cardiac muscle cells are rarely more than 150 micrometers long. Okay, so they're kind of a typical cell length. They're not very long. So we'll just call them short. And they also branch. That really makes cardiac muscle cells unique. They branch. Okay, instead of just being one long tube or a kind of a teardrop shape like smooth muscle, they're a branching tree. Okay, they have a trunk that goes out this way, and then they form branches. And that's important because when cardiac muscle cells are connected, they make a web. Rather than a long, thin fiber like you have in skeletal muscle, these branch out and make a net or a web. And that just has to do with how they function. When skeletal muscle contracts, it wants to make force across a joint, right? But when cardiac muscle contracts, it wants to point all that pressure down into a central focus point. So it's a globe of kind of net-like arranged cells. So when they contract, they force all that pressure down into the middle. So that's why they branch. At their branch points, where they connect, where two cells connect, they have a high degree of these special connections called desmosomes. And that's what we're showing right here. A desmosome is like protein stitching. We take proteins called fibronectin, and we use those to stitch in and out between the two cells, between their membranes, and then we have some proteoglycans that anchor them in either cell. And that makes a very strong connection. That's what a desmosome does. Back when you learned about the integument, you might have mentioned something like this when the epithelium sits on top of the dermis below. You take half of one of those, and that's what's called a hemidesmosome. And those are on the basal membrane of the epithelium where it connects to the dermis. Okay? It's the same basic idea there. Except here you have two sides where they go into both cells. So you find these desmosomes, which are these protein stitches that hold the cells together very, very, very tightly. So any tissue that's going to be subjected to a lot of stress, physical, mechanical stress, you're going to find desmosomes. And in fact, that seems to be what causes them to form. Okay? It's when we put the stress on the tissue that stimulates the production of these desmosomes. Another type of connection you'll find at this junction point is called a gap junction. Okay? You'll find these very large protein-based pores called gap junctions. Now this here is supposed to be the phospholipid bilayer down there. And I want you to understand that a gap junction is much, much, much larger than a simple channel protein, like a sodium or a chloride channel. These are 100 times bigger than that. These are very, very large. Gap junctions actually allow the cytoplasm of two adjacent cells to flow between the two cells. Okay, so that's kind of unusual. It makes the cells function together. Okay? Whatever happens in that cell over there also happens to me. Maybe a little bit later, but it's going to happen to me too. So gap junctions allow cells to share cytoplasm, which means that they respond in a coordinated manner. Okay? They respond as a unit. That's the function of a gap junction. It makes the tissues connected by gap junctions respond in a coordinated manner, is how we might phrase that. So if we were all connected by gap junctions in here, what that would mean is if somebody pounded Jennifer's hand, okay, Delara over here would go, ow. Okay? We'd all be connected. We'd all respond to that stimulus together. Okay? That make sense? It's really important for heart function because when we stimulate the right atrium via the SA node, we expect all the cells of the right atrium to get the message and to go ahead and start depolarizing. Not to hold out and be like, no, I'm special, I don't want to. We all need to respond together. Okay? And also, gap junctions can also be a special type of synapse. So back from EMP1, we learned about two types of synapses, and we spent most of our time talking about just the chemical synapse, the neurotransmitters and everything that did. But do you remember the other type of synapse? Okay. I don't know if Professor Rufisi talks much about it. I don't really in EMP1. But the other type of synapse is called an electrical synapse. And that's made by gap junctions. Okay. If you remember how action potentials are propagated by the influx of sodium into the cell. And the sodium diffuses inside the cell and depolarizes the membrane, and that's what creates an action potential. By having a lot of gap junctions here, the sodium is inside the cell. It can go through the gap junction into the next cell and cause it to depolarize. Okay? The more gap junctions you have, the easier it is for that to occur. So we call that an electrical synapse. The ions actually flow through the gap junction and create a stimulus in the next cell, an action potential in the next cell. So we don't have to have a cleft and the release of a neurotransmitter and so on and so forth. Can you think of some cells that might be connected via these electrical junctions inside the heart? Can we talk about something that this might apply to? Yeah, sure. Not just the SA node, but the whole conduction pathway. Okay. The molecular feature that is unique to those conducting cells is that they have a high degree of these electrical synapses, gap junctions. So that the SA node is connected to the internodal pathways, and then that's connected to the AV node, and it's connected to the bundle of His, and it's connected to the right and left bundle branches, and they're connected to the Purkinje fibers. And between all those cells, you have a lot of these gap junctions and electrical synapses, and that's what makes them unique. That's why if you stimulate the SA node, the action potentials travel very rapidly through that pathway. And yes, they do travel through the rest of the heart, but they do so more slowly. Does that make sense? Okay. That's how we identify the conducting cells. I mean, we can't really look at the gap junctions, but we know that that's what's there. Now, when you're looking at the histology slide, you cannot see gap junctions and you can't see desmosomes because they're too small to be seen in a light microscope. But you can see the thickness of where they join. And that at that junction point, we call that an intercalated disc. And so that's what you're looking at here. That's that black. Okay, those are intercalated discs. And you can see those in the scopes that I had set up. So I'll have those out again next week. So at the intercalated disc, you have these two specializations. You have desmosomes for strength, and you have gap junctions for coordination of response and electrical conduction. Anybody want any more of that repeated, or are you good?
Sure, repeat one more time, please.
Okay, the last thing I said, how we summed it up?
Mm-hmm.
Okay, so in intercalated discs, you have two specializations. You've got the desmosomes for mechanical strength, and you have the gap junctions, which allow for coordination and response and electrical conduction. All right, now we've peeled away the sarcolemma, that outer plasma membrane of the muscle cell, and we're looking inside. And you probably recognize some things here. We've got the alternating I band and A band, and those are striations. And you don't have to know anything about I bands and A bands anymore, except that that's what counts for the striations. We look inside the cell, and maybe we remember in a skeletal muscle cell, and I think that's what they're showing here, right? Well, they're showing connected, which is a little weird.
This is cardiac muscle up top.
Yeah, this is definitely a cardiac muscle cell. Huh. Maybe they're both cardiac muscle cells. But this arrangement, oh yeah, no, it is, okay, it's fine. So we're seeing these myofibrils are what we're looking at here. And within that we have the myofilaments, which are the actin are the little ones, and the myosin are the red ones. So those are our myofilaments, and those are the contractile threads. And they're surrounded by this stuff, which is the sarcoplasmic reticulum. So if you remember in a muscle cell, is kind of laterally enlarged endoplasmic reticulum. And its function in muscle cells is mostly to store what? Do you remember? What's stored in the sarcoplasmic reticulum? Calcium. Yeah, good job. Calcium. We store calcium in the sarcoplasmic reticulum. Now, in skeletal muscle cells, the ends of the reticulum are enlarged and they're kind of bulbous, and it's called the cisterna. In cardiac muscle cells, we don't so much see that. And also, the T-tubules, which are the invaginations of the sarcolemma, where it makes a tunnel. So you go along the surface of the muscle cell, and then it makes a tunnel. It goes down inside. That's the T-tubule. Those T-tubules are not as close to the underlying myofilaments as they are in skeletal muscle. And they lack the cisterna. So what that does is it slows down the rate at which action potentials are propagated, and it slows down the rate at which calcium is released. And that really is the only physiological adaptation I want you to understand. Calcium released into the sarcoplasm is the stimulus for muscle contraction. Do you remember that? Okay. When calcium gets into the sarcoplasm, it binds to troponin and kicks off that whole sliding filament model. Okay. That's true here too. But the difference is it's going to take longer to get the calcium to release, because the T-tubule is a little bit farther away from the sarcoplasmic reticulum, and the sarcoplasmic reticulum is more diffuse. It doesn't have those big cisterna. So our calcium burst into the tissue is going to be a little slower. And that's good, because cardiac muscle contraction, we don't want it to be as fast as skeletal muscle. We want it to be a little slower. Now everything is relative, all right? A skeletal muscle twitch is going to happen in about 2.5 milliseconds. Okay. A cardiac myocyte is going to contract over about 250 milliseconds. All right, so that's 1/4 of a second, which to none of us in here would we say 1/4 of a second is a long time, okay? But it's a lot longer than the 2.5 milliseconds for a skeletal muscle twitch. Skeletal muscles can twitch as fast as possible until eventually they run out of oxygen or ATP, okay, or maybe calcium, and then they stop. And that's fatigue. In which case, if it's just your finger twitching, your finger just stops twitching and you're fine. You wait a couple seconds and then it recovers. Also, you can also stimulate a muscle cell to the point where you've twitched it so fast that it cramps up and you get an unrelenting contraction. That's called tetanus, right? So in skeletal muscle, we can have tetanus and we can have fatigue. And it doesn't kill you. Okay? It may happen at an inconvenient time, but it's not going to kill you. In the heart, both of those situations are deadly. Okay? If the heart goes through fatigue or the heart goes through tetanus, either one means blood stops flowing and you die. So the slowing down of the onset of the contraction of cardiac myocytes is a protection to prevent against both fatigue and tetanus. And we'll talk about some other molecular aspects of that in the next lecture. Okay. don't want to